Hello and welcome, eager learners. In this third episode of our visual system series, we're exploring the primary visual cortex. If you have missed the first two episodes, don't worry, there is a wealth of knowledge waiting for you there. So if you haven't tuned in yet, take a moment to explore the previous videos. And now we're starting our journey into the primary visual cortex. So settle in, enjoy, and let the visual adventure begin. Last time we discussed the dorsolateral geniculate nucleus, in short DLGN, where the visual information is processed and passed on to the primary visual cortex. This transmission occurs through the optic radiation in primates. While rodents lack a distinct equivalent of optic radiation, the visual information still finds its way from the dorsolateral geniculate nucleus to the primary visual cortex. Often referred to as V1, the primary visual cortex serves as the first stage for cortical processing of visual stimuli. And in human neuroanatomy, V1 corresponds to Brodmann's area 17. Let's start with the anatomy and information flow. There are two V1s, one in each hemisphere. V1 is made up of 6 layers, with layer 1 being the most outward and layer 6 the most inward. So think of V1 as a building with 6 floors, but the top being the first and the bottom being the 6th floor. Now, most of the signals from DLGN go straight to the 4th floor, but some of them sneak into floors 6 to 3 and 1. In fact, the top floor might be a special spot for certain neurons that are good at telling which way things are moving, meaning direction selective neurons from the DLGN shell in mice. This way of organizing the connectivity in the 6-layered cortex is pretty standard, so that's the canonical information flow, but V1 has some exceptions from the canonical setup. One interesting thing is about layer 4. Unlike in some other brain parts where this layer has a mix of cell types, in V1 it's almost all made up of one type of cells, called pyramidal cells. The proportion of different cell types in different regions is just an example of how a detail like this can change the way the brain region handles information. And it's exactly those details that make things more complex and interesting in neuroscience. The thalamo-recipient layer 4 sends its influence directly to layer 2-3 through their basal dendrites. There, the visual information undergoes local processing. For example, this is where error signals arise. It means that our brain performs predictive coding, a bit like autocompletion in your text message, trying to predict what you will type. But in this case, we talk about visual input. If there is a mismatch between what you expect to see and what you actually see, the error signals arise. And this happens by comparing the bottom-up signals, so from the retina through the DLGN to the cortex, with the top-down, or in other words, feedback signals from higher brain regions to V1. After this fine-tuning, the information takes two routes, one through long-range connections to higher visual areas, and the other, a local transfer to layer 5 cells. From layer 5, the process continues, spreading information to the opposite hemisphere, layer 6 and subcortical regions. These subcortical regions are also targeted by layer 6, providing cortical feedback signals. In mouse, layer 4 is the first location where major integration of visual information kicks off. Unlike the DLGN's classic small round center surround receptive fields, layer 4 in V1 introduces a twist, elongated receptive fields, typically with parallel on and off subfields. Neurons with such elongated receptive fields are called simple cells. But where do these elongated receptive fields come from? Hubel and Wiesel, in their famous paper from '62, proposed that combinations of neurons in DLGN that have their center surround receptive fields aligned connect to simple cells in V1, giving rise to an elongated field shape. And this new mega cool feature of cells in V1 leads to the generation of orientation selectivity. What is orientation selectivity? The intuitive way to understand it is by reenacting the original experiment of Hubel and Wiesel. So imagine you have an electrode inserted in your visual cortex. This electrode listens to only one neuron, so it records the activity of one neuron. Now, I will show you a bright stimulus on a dark background, and what you will hear is the activity of this one neuron. The more clicks you hear, the more active the neuron is. So here we go. First, I move the stimulus horizontally, and you can hear that this neuron responds strongly no matter if I move leftwards or rightwards. Now, if I change the angle of the stimulus, the cell still responds, but much less. And what happens when I move it completely vertically? The cell doesn't respond at all. So this specific cell prefers vertical orientation, and that is because the receptive field is perpendicular to this orientation. So in simple words, the cell can see horizontal movement, but not vertical movement. Importantly, different cells can have different preferred orientations, but I hope with this example you understand the principle. As we venture deeper into the cortical layers, complexity intensifies. So by combining signals from multiple simple cells, the receptive fields get more complex, and these are called complex cells. It's time to reiterate the concept of receptive fields, specifically to understand what eccentricity is. Imagine your retina as a detailed map where each tiny spot known as the receptive field captures a piece of what you see. The eccentricity of these fields indicate how far they are from the central part of the map called the fovea, where your vision is sharpest. To even better visualize it, picture the retina as the city and the map of the fovea resembling the city layout, with the fovea as its vibrant core. Now consider receptive fields as neighborhoods. In the retinal city, the size of a receptive field is described in degrees of visual angle. In the proximity of the fovea, the city center in our analogy, there are small neighborhoods resembling cozy little houses, measuring merely about 0.1 degrees in size. But as you move towards the outskirts of your vision, more eccentric, those neighborhoods become more spacious, stretching up to 10 degrees. This architectural shift is why areas near the fovea capture high detail visual information thanks to their small receptive fields focused on tiny portions of the visual scene. If it's further away, more eccentric, the receptive field gets bigger, so they capture bigger chunks of the visual scene. The consistent pattern is that receptive fields with the same eccentricity are relatively small at early processing levels and become progressively larger at the later stages. Another important term is the cortical magnification factor. It captures the relationship between the distance of 2 points in a visual scene and their corresponding representation on the cortical surface. Importantly, the central part of the visual field commands the largest area of the cortex. For example, in V1, more area is dedicated to the central 10 degrees of the visual scene than to all the rest. It means that the majority of processing is focused on the center. It is a bit like a fisheye lens where the center is blown up compared to the squashed periphery. With these basic concepts covered, we can focus on seven crucial aspects of the functional architecture of the primary visual cortex. These are the retinotopic organization, ocular dominance, orientation selectivity, direction selectivity, spatial and temporal frequency preference, and color preference. Let's start with the first one. The most prominent feature of the primary visual cortex is that it's retinotopically organized, or in other words, visuotopically organized. It means that the primary visual cortex is like a finely organized map in your brain. Every point on this map corresponds to a specific location in your field of vision. So imagine your visual field as a big canvas, and the primary visual cortex meticulously represents this canvas on its surface, with the center being blown up as we discussed a moment ago. The second important term is ocular dominance. The primary visual cortex consists of monocular and a binocular region. What does it mean? In animals with binocular vision, there is a partial overlap in the visual fields observed by each eye. How big this overlap is depends on how far apart the eyes from each other are. For example, humans have a high degree of binocular visual field overlap, roughly spanning 130 degrees. That's because humans have their eyes positioned in the front of their head. On the other hand, mice, with their eyes on the sides of the head, have only 40 degrees overlap. This shared or binocular visual field finds its representation in the cortex within the binocular region, where inputs from both eyes are processed. And this is super useful for depth perception because based on the difference in an object's position between both eyes, we can judge how far it is. Within the binocular region of the primary visual cortex, each individual neuron exhibits ocular dominance. It means that a neuron receives an input from both eyes and it responds more strongly to one of them. To capture this phenomenon, scientists use an ocular dominance index, in short ODI, that ranges from -1, where the neuron responds purely to the ipsilateral eye signals, to 1, responding purely to the contralateral eye, with 0 being perfectly binocular. In some mammals, including humans, these binocular neurons with different ocular dominance are arranged in columns. These columns span across all cortical layers, and they are believed to arise from segregated input of different layers of the DLGM. So in the cortex, we have alternating left eye and right eye dominant bands. We already discussed orientation selectivity earlier in this video, but what's interesting is that Hubel and Wiesel noticed that neighboring cells in the V1 tend to have the same preferred orientation. They later found a continuous columnar progression of preferred orientation along the cortical surface. These maps of preferred orientations were visualized in great detail and shown to be arranged in pinwheel patterns with orientation centers. It means that when looking at the cortex surface, the preferred orientation of cells continuously changes in a pinwheel fashion around the center of the pinwheel, while in the center, the orientation suddenly changes. Additionally, there are functional maps of preferred direction and preferred spatial and temporal frequency. Similar to the orientation, neurons respond strongly to their preferred direction of a stimulus. So, if we recorded the activity of a direction-selective neuron, it would respond to movement of the stimulus only in one direction, but not in the other. Cells also have their preferred spatial frequency. It can be easily illustrated by showing visual stimuli with different thickness of black and white stripes. These kinds of stimuli are commonly used in visual neuroscience as they evoke the strongest response in the brain. Some cells also care about how fast these stripes move, and these are temporal frequency selective cells. Interestingly, the superficial layers within V1 house clusters or blobs of neurons selectively tuned to color. This intricate architecture of different features is thoughtfully arranged, and it has been put together in comprehensive, overlapping but independent topographic maps at the turn of the 90s and 2000s. Most of the just mentioned features were discovered and confirmed in cats, ferrets, and primates. In rodents, however, these neuronal representations of preferred orientation, direction, spatial and temporal frequency, and ocular dominance are believed to be randomly scattered. It's often called a salt and pepper organization, but recent studies suggest that the salt and pepper map might not truly be randomly arranged. Studies have found hints towards possible micro and macro organizations in rodents, such as ocular dominance patches and a global map of orientation tuning. Okay, these were a lot of maps. Let's recap what we have learned today. We started our exploration by tracing the information highway from the dorsolateral geniculate nucleus to the primary visual cortex. Delving into the anatomical architecture of V1, we discovered its 6 layers and the canonical information flow in the cortex. Moving forward, we explained the concept of receptive fields, envisioning the retina as the detailed map capturing various degrees of visual detail. Understanding the cortical magnification factor, we recognized the central part of the visual field's dominance in the cortical space, similar to a fisheye lens effect. Our exploration then focused on 7 crucial aspects of V1's functional architecture, covering retinotopic organization, ocular dominance, orientation selectivity, direction selectivity, spatial and temporal frequency preference, and color preference. Each of these elements contribute to the rich variety of V1's topographic maps, meticulously arranged for optimal processing. This way, the brain ensures that all combinations of features are extracted from all retinotopic positions. I hope you enjoyed watching this video and that you understand the complex architecture of the primary visual cortex a bit better, but our journey doesn't stop yet. We have one more exciting video ahead looking into the higher levels of visual processing, where the complexity of perception continues to unfold. We will talk a lot about optical illusions, so stay tuned and I look forward to continuing this journey with you.